afternoon, everyone. Welcome to an Australian Institute of Health Innovation seminar with Professor David Goldstein, who I will introduce shortly, and the topic. So, as you know, many of you come to these. This is a series where we have experts from within and outside of the Institute give a talk on an area of their expertise and often an area of their passion. And today we'll certainly be hearing somebody with oodles of expertise and high levels of passion for the topic we're going to hear him speak about. So the title of this talk is From the Bench to the Bedside and Back. So I often hear about from the bench to the bedside, but I'm very interested in going back to the bench, with the TCRN as an example of fostering translational research. Just a quick advertorial. The TCRN is the Translational Cancer Research Network that David heads, and it has been going now for almost 6 years, looking at how do you get evidence into clinical practice in cancer in several teaching hospitals on the eastern seaboard. So the TCRN is based at UNSW, University of New South Wales, and funded by the Cancer Institute of New South Wales, and several people in the Institute who I can see in the audience are members of the TCRN. So the idea is to foster a sustainable culture of translational research to do with cancer, on oncology services, and to demonstrate how that might be done in partnership with many stakeholders. The speaker is Professor David Goldstein. David, welcome to the Institute. It's a pleasure to have you here today. David's conjoint clinical professor at UNSW at the Prince of Wales Clinical School. He's well known in cancer circles and also has managed to straddle those two impossible jobs, being an excellent oncologist and an excellent researcher. And it always strikes me that there are still people who are prepared to do two impossible jobs and carry them off with aplomb, and David is one of those. So he works especially on the treatment of GI malignancies, including pancreas cancer, colorectal cancer, anal carcinoma, and upper GI malignancies. And he's very interested also in renal cell carcinoma and lymphoma. So these are areas of expertise. But really, he stepped out of that role to some extent and headed up the Translational Cancer Research Network. And I'm proud to be part of the team that he leads as a member of the TCRN. So that's what we're going to talk about. That's a little about who's going to talk to us. Could you please welcome Professor David Goldstein?
So just some background to start with. So what does all this mean? The Cancer Institute back in 2011 was getting really a very disparate set of applications for grants and for funding from all over the state, and it had been set up to foster research in cancer and actually to improve service modelling in cancer in this state. So they thought perhaps if they funnelled and aggregated requests for grants through centres that were logically placed and surrounded by universities and teaching hospitals, that might result in greater gains, increased synergy, increased interaction between people. So they nominated 7 translational research centres across New South Wales and invited bids saying, well, if you got nominated and you got funding, what would you do? And usually they were looking for partnerships between the local health district and the affiliated university. And a network was to be membership-based, so it wasn't just to appear out of nowhere and have no credibility. So the Cancer Institute had its own objectives and themes. It was about fostering translation and innovation, and it was about building capacity. They were the two overarching broad ideas. And they come from a background that you know much more about than I do, which is this idea of the T1 to T3 spectrum, from very basic research moving into human clinical research rooted in that basic research, so phase 1 and 2 clinical trials, some of which end up being phase 3 to prove practice changing, with associated health research studies, and then moving into policy and practice, looking both at delivery and systems of delivery, and also trying to identify through meta-analysis, systematic reviews, and the process of translation, because as many of you exercise your minds on this all the time, just because there's evidence that something works, it doesn't necessarily mean that it gets taken up in practice, and trying to find out why not is a key part of trying to improve outcomes, in addition to the first bit of work to show that something new actually does something different. So the previous leadership of the first 5 years of the TCRM was Professor Robin Ward, who's moved on upward in the academic hierarchy by going to the University of Queensland, and supported by a team of back office people that are the engine room that makes things work. So Robin's vision, which we've tried to carry forward but with some changes, some of them imposed upon us, in fact, as I'll explain later, was to foster a sustainable culture of translational research. So that means from basic science influencing clinical service delivery. Probably not going to spend the time I should on the reverse, but now that it's been brought up and I realise I haven't focused on it much, I might towards the end discuss that. It's about a sustainable cancer-focused workforce, to identify areas that would benefit from focus and change. What's the problem? And test and develop pathways to address these changes. So that sounds great. Wonderful ideas. Question is, what the hell to do, as opposed to what sounds like a magnificent idea set on high from our leaders at the Cancer Institute. But the group, of which I was one committee member, kind of did translate into reality. So if I was to give you a snapshot, and you can get up and walk out after looking at this, 5 cancer challenges of the year were articulated. What does that mean? Is there a problem out there in a particular aspect of cancer care? And can you address how to deal with that problem encompassable in 1 year and $100,000 of project support? And I'll show you, the crux of the talk is to show you examples of that. In addition, a biobank was established. But the idea of this biobank wasn't to just get another researcher who's interested in breast cancer to collect 200 specimens and do something with them or just collect them. It was really about, could you make the process of biobanking a routine part of a public hospital's activity, and in doing so get a very rich collection of specimens, but also get appropriate information to match those specimens so you can start to ask clinical questions that influence practice. So that's the second half of the activity, which was translational bioinformatics, and I'll come back to that. So if we think about the broader plan of building a workforce, then there were top-up scholarships, and 31 very diverse PhD students across the network were funded. There were first author publications as a result, and there was a fair bit of money spent on supporting people to go to conferences and professional development. A few minutes on our network, which I didn't do, that I should do. So for our particular centre, University of New South Wales, University of Technology, Sydney, and one group at Macquarie, your group, were our partners, together with Prince of Wales Hospital, St George Hospital, and a large regional cancer centre in Albury-Wodonga, Border Medical Oncology. So the other thing to state is that a concept called the Academic Health Science Alliance was part of this process, which was really replicating well outside cancer, but that idea of the nexus between a university and university researchers and clinicians working in areas that those researchers are interested in. So I want to just give you an initial flavour of the cancer challenges and drill down to a few, just to give you an idea of the fact that they were very focused. So the first one was actually about pain management and earlier recognition by frontline nurses of pain needs, and as a result, earlier treatment, earlier discharge, better quality of life for cancer patients who were brought in either because of pain or for some other reason, but pain was one aspect of their care, using an adult education technique called spaced education. And that was undertaken in 7 wards across 5 sites. The second, which I'll go into in detail, was a new screening technique for Lynch syndrome, hereditary bowel cancer, using a simple immunohistochemical test. The third was trying to address the challenge of how to engage family members when one index person has been found to have an inherited cancer, because engagement of family members is often very difficult, but the leveraging of reduction in risk requires that you reach out to the family members. It's not just enough to identify the person who already has it. It's the snowball effect beyond that. And the development and pilot testing of a talking book to facilitate for patients with low health literacy who are undertaking radiation treatment. And then another one with a familiar face was achieving behaviour change for prevention and management of Lynch syndrome. And I'd be very remiss if I didn't make that the main focus of the whole talk. In addition to coming from our members to us for PhDs, for cancer challenges, for going to conferences, there were some ideas that came out internally. And the first, which I'll cover later, was an attitudes to cancer follow-up care. The second was working to improve a laboratory process, that is next generation sequencing for inherited breast cancer. And the final one was really through Jeffrey's intervention, an attempt to understand metrics. How do you judge if doing all this stuff is useful and achieves more than would have happened anyway? And I think that's very challenging, and I welcome further discussion about that towards the end. And then another significant initiative was a consumer advisory panel, one of whose members is actually here today. So really, there's been an increasing trend across cancer research in this country that ensuring relevance by engaging the people who are directly affected by the disease or the spectrum of diseases and the outcomes increases the chance that you will end up with research that will have broad applicability. So the Consumer Advisory Panel has served a variety of functions. It's provided feedback on research projects. The NHMRC now expects that a consumer's been involved from the generation of the idea through to the planning of the implementation of the research, as well as looking at the outcomes. There's been a number of one-on-one where a consumer is affiliated with a particular, embedded now in a particular research group. Input to the TCRN. So at each one of our management structures, we actually have embedded consumer input right from the top of governance to the granular individual research projects. So we have a cast of, I think, 14 now rather than 10. So I don't have, the Rogues Gallery should be longer and will be in the future. And they're all very engaged in the process and very articulate actually and helpful. So we have 220 members. So as I said, UNSW, our particular Cancer Research Centre, UTS, and the Royal Hospital for Women, as well as Prince of Wales, St George and Sutherland, and Border Oncology. So I have many masters and I have to go and meet them all the time. So it means I never get to do anything except talk to people anymore. That's an interesting challenge in itself. What about trying to ensure diversity? So I think we've actually done a good job of that. There was diversity in the previous group, and I think we've built upon that. So there's me, I'm a medical oncologist, so I'm the director. There's a deputy director who's an academic general practitioner running a general practice research unit at the university. We've got a haematologist who's really quite a renowned laboratory researcher, but also an active clinician, and he's involved in brokering translational activities. A director of area cancer services who's also a pharmacologist as well as a medical oncologist, an academic surgical oncologist, an academic nursing researcher at UTS, and you know who Jeffrey is, and in terms of future proofing, we now have 2 ex officio members, medical oncologists, Catherine Soquist at St George and Kate Webber at Prince of Wales, both of whom have recently finished their PhDs as well. So that's kind of looking towards sustainability and the future. And what about our governance then? So the way we've set it up, we have a scientific leadership group, that's the people whose pictures you just saw. Below them and answerable to them, like in any good organisational structure, is a small executive who are the workers. Myself, the Deputy Director, Translational Broker, Director of Cancer Services, and Stephanie McMillan, our Program Manager, is sitting in the front here and actually does all the work and takes all and should take all the credit for our success, but also our failures, if we have any. I'm going to explain to you what this term flagship is in a moment. We are just about to start what we call a scientific advisory group. Its acronym is SAG, which worries me, and we're going to have to work out how to do better than that. But they're just going to twice a year give us a bit of perspective and feedback about whether they think we've got a reasonable direction. And so you can see all these people I have to meet with twice a year, the universities, UTS, UNSW, the Southeastern LHD, and the leadership group, as well as the 2 hospitals and the regional centres. So already the calendar's full before I've started to do any work. Now, what about where I came in? So Robin Wood was leading it, she's moved on. The Institute said, look, this wasn't bad, we aggregated stuff, we're quite happy, but we want to do even better. We want to challenge you. We want you to look back at the last 5 years, tell us what you think your strengths were, and identify flagships that will be the main engine room of research within your translational, rather than everybody doing everything in all our 7 centres. So we thought about this a bit, and have come up with hereditary cancer, what we call continuum of care, which is engagement, community versus hospital, clinical improvements into care looking at haematology, surgical quality, multidisciplinary care, and biobanking and bioinformatics. So stuff that we already felt we were good at, leading in some new directions, and some other things that perhaps because of partnerships we thought we could do well at, even though we hadn't done them before. So what I'm going to do is just go through each flagship and give you a flavour of why we felt confident in putting that flagship forward. So the first is hereditary cancer, and its main goals are dealing with a volume of work that's too high for the workforce to cope with by looking at novel ways of referral and management, video conferencing, web-based, some patient-driven, some referrer-driven. So what led up to this? So I think the first one I want to talk about is this screening for Lynch syndrome. So Lynch syndrome, about 3% of all cases, but the key is testing of first-degree relatives to identify additional people at risk and prevent them getting a cancer, and secondary prevention of new cancers. So colorectal cancer, there's another 1% per year risk of a second cancer, and that's much higher in a Lynch syndrome group than it would be in the population. And there's a simple immunohistochemical test done in most anatomical pathology laboratories in every teaching hospital in the country that's able to identify mismatch repair. So the hallmark of the potential for an inherited cancer. However, 90% of these patients won't have Lynch syndrome. There'll be sporadic changes in the tumour that aren't reflected in the host genome. So this additional mutation, BRAF, occurs almost exclusively in the sporadic non-hereditary type. So if you could test for BRAF mutations easily, you could enrich the population you identify that should have the much more expensive and detailed genomic sequencing to really drill down and understand what this hereditary tendency is and how to look for it. And so Nick Hawkins, the professor of anatomical pathology at the university, identified a new BRAF mutant antibody and a simple screening tool to pre-screen and tested it in the southeastern area anatomical pathology departments. So he found first it had a positive predictive value of greater than 93%, so that's looking at the gold standard of NGS, that the proportion of patients tested that didn't need referral was somewhere between a confidence interval of 50 to 85%. So in a really busy hereditary cancer clinic and a very busy genomic laboratory, halving, or maybe more than halving, the volume going through is clearly going to have a major impact on service delivery and a focus on dealing with the people who need something done and not spending time you don't have on those who don't need anything. Clinical deployment, however, required thinking through what is it that will be an incentive to a busy anatomical pathology department to do yet another test, which won't get paid for, which will take time, and meanwhile, clinicians like me are saying, have you got the result yet for my patient who got operated on yesterday? I want to know what chemotherapy to give them tomorrow. So working out the facilitation that will make the evidence translated into practice was what Nick Hawkins, as an active card-carrying anatomical pathologist, was able to think through, and a tech transfer package to reduce the pain of doing this next test. The other side of the coin was this. So Natalie Taylor, who you know well, led a team that was asking, is everybody at risk being referred? Because there was a suspicion that they weren't. So a 6-step method that I am simply too intellectually inferior to understand, but is called Theoretical Domains Framework Implementation. I confess I know nothing about it, but it sounds great. I have a black box theory. I don't really mind about what's going on inside the box and the wiring, as long as it leads to an endpoint that I can understand. So I did understand the endpoint. But it involved collaborating widely with clinicians, pathologists, nurses, genetic specialists, data managers at 2 large Sydney hospitals. And what they did was a straightforward but necessary process. What are the processes around referral? How does it happen? And what are the issues around that? And what did they learn? The referral rates were dreadful compared to what they should be. And why? Everything you can imagine, it's the whole system problem. Environmental context, resources, social influences about, have I got time to refer, and do I really need to, and is it important? And the skills, right, of the different people involved, and maybe some belief about capabilities to identify. So changing practice, it became clear, wasn't just about the individual surgeon who'd done the colon cancer operation. It was about a whole bunch of other people and their ability to do their piece of the puzzle that leads to the identification of the at-risk patient. They found that data always works. If you show 36% referral, people look up and say, that's a number we're not proud of and we need to change. Leveraging social capital. So if you get a surgeon speaking to fellow colorectal surgeons, has more credibility than someone else saying we need to do better than 36%. The TDFI approach is effective, feasible and acceptable. That's good. Simple people like me might be able to use it. It's led to a number of collaborations. It got onto the Twitter sphere, I understand. But most importantly, it got funded. So this is absolutely what we're supposed to do. Facilitate something happening, Cancer Challenge of the Year. It provides compelling data and leads to writing a grant, and the grant gets funded. So let's turn to a different one, continuum of care. So Mark Harris, academic professor of general practice. So what are his plans for the next 5 years? They're pretty ambitious. Develop and evaluate the implementation of primary health care engagement models across the continuum of cancer care and information systems, these so-called web-based health pathways that lead back into the hospital and from the hospital out into community care, so that the information that's buried in the hospital can be accessed the first time the cancer patient turns up in the general practitioner's office saying, well, I've been under treatment and follow-up for the last 5 years, so I haven't felt I needed to see you, but Goldstein discharged me from the clinic today, wished me luck and said I don't need to see you anymore, so I'm here. And now the general practitioner has to unpick what that means. What are the potential implications in follow-up and management? What might be the survivorship issues because of the type of treatment? And how do I deal with them? So that's sort of where Mark Harris is going with this project. Why does he think he can do it? Because he did this Attitudes of Cancer Follow-up Care in General Practice. So all of our clinics are getting significantly loaded up with patients in follow-up. So the question is, how acceptable is it going to be to providers and patients to change that model into a general practice-focused model of follow-up and care? And so a multidisciplinary group, oncologists, academic general practice researchers, nurses, was set up, and what they did was, I understand, an almost unique study. So they actually matched the patient's oncologist and the patient's general practitioner and asked all three the same set of questions about follow-up, its complexity, and what they found were the barriers to doing that follow-up. And they focused on breast and colon cancer survivors with no current evidence of recurrence. That's the group to move. Can I say that people like me are moving ahead of the evidence? Well, there is evidence, so using the evidence we know internationally and are rapidly transferring patients either to the surgeon or a shared care of an assumed successful shared care model of GP and surgeon, with me moving out of it completely just because I've got more and more new patients I need to deal with. And the success of what I do means there's many more patients having second, third and fourth line cancer treatment in a palliative sense, and they're living longer. So I no longer have any space for follow-up in my clinics anymore. I can't do it. So I'm just sending them. The problem is I don't know whether the dots are still connected. I'm just making a leap of faith that they're connected. So that's what Mark Harris is going to do. He's going to show that the dots are connected and I can step out. But getting back to this triad and what they learnt. So 22 patients, 18 GPs, so they didn't get everybody. Nevertheless, what did patients say? And this is very consistent across all the research that's been published in this area, is that patients are uncomfortable being divorced from their oncologist. They are wedded. They've been through 5 years of treatment and follow-up. They are not happy at the idea that they have no communication anymore with the people that have followed them on that journey for so long. But so they wanted the GP to be part of, not instead of. That's a very clear message. And we have to design things that will make them comfortable. Parenthetically, what patients don't necessarily appreciate is that many of the survivorship problems, hypertension, obesity, exercise, are better done by general practitioners than by people like me. So there's a need to recognise. So GP said, we don't know about cancer very much because it's 5% of my practice. Diabetes is what I do every day, and hypertension. They wanted protocols. They wanted clarity about what to do and when to do it, right? And they wanted access to oncology teams. Some oncologists didn't appreciate what the role of the GP should be. Others were very enthusiastic. Others, like me, had already done it without working it out properly. So defined roles, protocols, the routine elements of follow-up care that don't require a busy oncology clinic to do them, and making sure information sharing and the passage in and out of the hospital when you get a problem that might smell like it's a cancer problem or a very specific toxicity that needs the attention of somebody who's experienced with that toxicity. So that's really why we felt we can do this. That's why we're going to say that's another flagship for us. I should add that the other thing is we've had a survivorship centre actively involved in this for the same time that all of this is going. So we had a number of elements that justified that. What about biobanking and bioinformatics? So we've said we're going to enhance and improve research output, data capacity, bioinformatics, infrastructure, improve next generation sequencing, and pilot this idea of a molecular tumour board. So that is a rare cancer, trying to identify the right treatment for the right patient at the right time is what the new personalised therapy approach is, and that means understanding the molecular makeup of the tumour and then planning the treatment. Much more complicated than it sounds, but we're going to pilot a molecular tumour board so that when patients are faced with, and their treating clinician with no obvious treatment but a molecular set of tests, what do we do about it? I can't see time here. How am I doing?
22.
Great. Okay, so what's different about a biobank? The first is a biobank where all patients have the opportunity to contribute, where the process of consent are part of routine care, not some special deal. This is just what we do. And finally, that the biobank is completely open to external researchers. We don't somehow close it up and it's only for us. But we do look for partnerships. It's not just you can have it and we don't really mind. The patients agree to it with their researchers in mind, as well as being altruistic to the whole world. So it took 18 months to go through the forms committee. The patients want it, the researchers want it, the treating doctors want it, but the privacy laws and hospital bureaucracy prevent things from happening. And I have no idea how to break through that problem, but it's probably the biggest problem facing global people like you who want to do global health research and health outcomes, is that the barriers to you getting the information to ask the questions are enormous. And this is one hospital. And all we wanted to do was to give patients the opportunity to say, yes, you can have a sample of my tissue, and yes, you can interrogate the MBS and PBS data about me afterwards. And yes, you can take everything that sits in these disparate silos inside the hospital about histopathology, about blood tests, about patient letters, about anatomical pathology, and aggregate it together to understand something about outcomes. So they got through that 18-month process and did well enough that in fact now New South Wales Health Pathology for the whole state has adopted something similar, that sort of consent form. So it'll be a statewide initiative to biobank. I think it's going to take 1,000 years to successfully go through the next set of bureaucracy of doing it statewide, and probably 20 millennia to do it for the country. But I exaggerate a lot, but it's going to be a lot of work, and it isn't going to be available for this group to interrogate next year. So what do patients want? Well, all this stuff about, well, they're going to be under duress, they're thinking about the operation they're about to have the next day, this is an unreasonable burden, and you've got to go back a month later and check that they really wanted to do this. 100% of the patients said, this is a great idea, and no, I'm not worried about the fact that you approached me before surgery to get me to agree to it. It's not an issue. So what did we get? We got really quite a wide range of different tumours. Some, like gynecological tumours, got a high percentage, others a low percentage, and that inevitably is a result of the enthusiasm of the team rather than the enthusiasm of the patient. I'll come back to that in a moment. But here's the very difficult issue. 5 years of work and less than half of the samples, less than a quarter of the samples, do we have all the matching clinical information. Despite a New South Wales Cancer Institute funded clinical cancer registry, despite a New South Wales state cancer registry, and despite clinical information being warehoused within hospitals. The complexity of bringing those together means we only have 653 samples where we know everything about the patient. We know about baseline demographics on everybody, let's be clear, and about 1,400 where the presence, the unique ability to access MBS and PBS data gives us greater capacity to have matching clinical data. So that's why we were able to confidently say that our focus is about getting access to the data in real time and in a semi-automated fashion. We actually have to get rid of eyeballs. Manual extraction from clinical charts of data is an impossible situation that cannot continue. It's not sustainable. It can't be done. That's why we've only got 653. We've got to find some other way of doing it. So that's our task. So what did we learn? We learned that when it was offered, 95% of people agreed to do it, but only 45% ever got offered the chance to do it. And that's because we have a just-in-time system in hospitals now. So it's not like they come in the day before to get prepped and you can have a chat. They come in at 7 o'clock in the morning, they're operated at 11. Preoperative clinics are now, because of health services research, triage, so only people with significant comorbidities end up going to a preoperative clinic where you might have access to get a consent. So there are technical problems that we've learned. Changeover, an enthusiastic surgical fellow, one lot of 3 months, less enthusiastic, much more interested in learning how to cut than get a consent form for tissue, the next 3 months. Enthusiastic surgeon who wants to have a biobank to interrogate with their partners in the basic science area versus one who's really engaged in good surgical service and quality and not in basic science research. These are all of the issues, and sustainability. And the other half of the coin is what I've been banging on about, translational bioinformatics, getting the information to match the specimen and the science. And a lot of good work has been done trying to find mechanisms and applications to do that. So that's included meeting privacy legislation and anonymity that will satisfy hospital ethics committees about transferring the information out into the university to match it with the specimen that's been collected, and then getting that information but putting it in a way that's interrogatable. So that's what Open Clinic is, one of many systems, but it allows you to interrogate a disparate group of patients with disparate information. Very interesting is imaging data. We got a request recently from a group in the US who believes that they can work out prognosis just by looking at a blown up single H&E stained slide of a patient and can predict their outcome. Watch this space, but it's kind of interesting. And it led to a lot of interest externally. You know, we're not the only people doing it. Some of members of your group are collaborating with our bioinformatics, but also with plenty of other people to try and sort out the problem. So one of my mentors and friends, John Salzberg, says that a biobank without clinical information is a bank that pays no interest. So building on that, I thought we'd talk about what we've done that gave us the confidence to put this up as a flagship, in addition to the fact that the mechanics of it was accepted as state-of-the-art mechanics. And one of my colleagues, Nikki Ma, led a project where 170 biobank colorectal, breast and ovarian cases were looked at that had some clinical information, to ask the question, let's forget about the hospital. If we've got this MBS, PBS, will that be a surrogate that will give us everything we need to know that we seem to have trouble accessing from the hospital? And to a lesser extent, yes, it improved accuracy of primary diagnosis, which was backed up because we had one anatomical pathologist working half-time, looking through report after report and then trying to give a single accurate primary diagnosis. So that was helpful, and it was an efficient way of determining disease progression and recurrence, because you kind of look at no treatment, suddenly they're getting chemotherapy. That's a surrogate date for when they recurred, for example. And you know, types of investigations. So another project that was done was there are guidelines about types of investigations like CT scans that should or should not be done in early breast and bowel cancer. So you're able to use this kind of data to examine whether there's over or under ordering of appropriate tests, right? And the PBS can tell you about which drugs are used and not used. And I was only talking to one of your colleagues this morning about using that as a way of examining real-time outcomes in the real world as opposed to the phase 3 result. Surgery is a bit of an issue because the MBS can pick up the private surgeries, but you need the public hospital registry to pick up the public surgeries, and we're doing some work around a clinical cancer registry to look at surgery in upper GI and gynae as a result. And the second is hot off the press, just funded by the Biobank Stakeholder Network, is a partnership that will involve your group, and it's looking at exactly that, trying to increase the use and integration of these disparate information systems in hospitals and work out ways for timely semi-automated data capture that reduces the number of people in the chain of information acquisition. And I presume that has spin-offs across the entire health system and not just cancer. We're just trying to work out the way to do it. So there's a minimum data set that previous work identified. And it happens that there's 2 existing manually created clinical data sets where that information's been collected by hand, laboriously, over many hours and weeks and months of work. So try and work out a different form of electronic data capture and then compare how it performs with the previous manually created work is one part of what that project's about, and the other part is the IT system. So lastly, this clinical improvements into practice. I'm not going to talk about quality of care in surgical oncology, but that's one of our goals. We're looking at enhancing multidisciplinary care, and I won't talk about that today. I just want to talk about the haematological malignancies. So I have 2 really innovative haematologists. One, Mark Hertzberg, looking at acute myeloid leukemia. The other, one of our, or the national ranking expert on myelodysplastic syndrome. We're also doing some big work on thromboembolism by an international opinion leader who works at our hospital. I won't talk about that today. But where did our confidence come? Nobody else in New South Wales has put up haematology as a flagship. So where did we get the impertinence to think we should do that? So it came from a couple of things. First, that because we're a reference laboratory for genomic sequencing, both for the Sydney Children's Hospital, because it's very important for childhood genetic disorders, and also because we are one of the 2 reference centres for hereditary cancer in the state, one at Westmead for the northern half and we're the southern half. So because we co-invested in a fellow who worked inside the lab, they were able to improve sample processing and reduce cost for BRCA identification. But from the haematology point of view, a diagnostic approach to acute leukemia that looked at next generation sequencing and assays for particular genes that had prognostic implications. So the next step is to be a reference laboratory for haematologists to match treatments with the initial genetic makeup of that particular leukemia and test whether that influences outcomes. And the same is going to be done for myelodysplastic syndrome. So lastly, all of this is kind of moot if you don't have people who can utilise research and if you don't have people who can drive new ideas. So we have this silo called the 2025 Cancer Workforce, and that's to develop the next generation of researchers and clinicians through undergraduate, postgraduate programs, translational brokering, and that is where I'll digress for a moment about bedside back to the bench. So it's really about integrating clinicians into research programs. So for the last 20 years, I've been working with first 1 and now 3 different laboratory programs where I front up every second week for an hour to an hour and a half. And as part of the routine laboratory discussions that happen every week, how are we going, this research, this particular experiment, what did it show? And my role is to give a clinical perspective about the relevance of what that is to how I treat a pancreas cancer patient today and what the next phase 2 and 3 trials that are coming down the track are and what the animal modelling that would benefit me should be. So we want to try and triple and quadruple the number of people who do that kind of thing. So I'm pleased to say in our centre, gynae, oncology, particularly ovarian cancer and brain cancer, has similar people doing exactly that. And the reverse is that we're going to get the scientists to come to our clinical meetings every week where we present our new patients and how they're treated. And the gynae oncology group has been doing it for 5 years, and it has influenced the questions the researchers ask. So we're going to do more of that Project of the Year funding, but it'll be targeted to the flagships. We've got some PhD top-ups. We've got this new idea. It's very hard to get clinicians to give up 3 years. They've spent 6 years becoming a surgeon. Are they really going to take another 3 years off to do research? Usually not. But we have some incentives that we're going to do to see if we can change that. And we've got some dedicated cancer APAs, conference and professional, and as I mentioned before, the consumer advisory panel, just a flavour of the kind of courses that we've done. But I reflect on facilitation. So I've talked about a whole lot of research. I haven't done a single one of those things. I haven't been involved in a single one of those projects, but I've been a facilitator for them. So one asks, what sort of facilitator? Should you be the great facilitator? Is it the great helmsman, Chairman Mao, leading the country forward? Or is it the guy who says, I want people to be afraid of how much they love me from the office, right? No idea. But there are some components of facilitation that industry models talk to us about. And it's about these sorts of things: create meaning, manage the process, take responsibility, ask questions, be conscious of the power of facilitation. I've got no money to give. I'm not the NH&MRC. The TCRN doesn't offer $600,000 a year for 3 years as a project grant. All we do is a little bit of seed money, a lot of facilitation, a lot of brokering and bringing people together. That's all we've got to offer on our budget. So that leads me to this final question. How do we prove that a translational research centre does anything useful? So it's pretty obvious. Let's take all their grants. Well, how do you know they wouldn't have happened anyway? All right, let's take the ones where more than one research group is working together and say, well, somebody brokered that. Maybe it was us, but you can't prove it was us. What about publications? Same deal. What about the composition of research groups? Are there clinicians attending the lab program? Well, I went to my lab program long before there was a translational research centre. How do I know it would have happened anyway? Well, if suddenly there are 50 of them when there was only 3, I guess you might be able to say you've done it. There won't be 50. I think if we get 10, we'll get a gold star on our foreheads. External educational activities, and ones that we've done, and I won't go into, but we've done some, we've got more. So your institute came to our rescue and they said, we have a brilliant thing called social networking. We're going to answer and prove it. So they invited TCRN people to complete an online whole network survey. They did also some qualitative research, and I've got about 70% of people. And the most important thing is not the number of collaborative interactions in that forest plot, network plot you saw, but more that 40% of the ties were with people they didn't know before the TCRN started, and a third could list projects not funded but attributed to being inside the tent. So the alarm was that only 4 people had the vast majority of the interaction, the largest number of interactions, and that's not really sustainable. If all 4 people move on tomorrow, what happens to the network? So it's just something to remember. These are the people who make it happen. It's not the director or the deputy director. It's actually the human infrastructure. But that costs money and salaries. So people look at me and say, look at your budget. It's all spent on salaries. What are you doing? Why aren't you giving it all to the researchers? Well, if you want to facilitate, you have to have a program manager who understands the whole program and the spread of resources. You have to have people who examine metrics. You have to have a bioinformatician. You have to have somebody who understands about project management, and you have to have people who can work 1 million calendars and bring all these busy people together in order for projects to be done. So I'll stop there and entertain some questions. Essentially, that's what we do. We have a full-time bioinformatician who's just completing his PhD in bioinformatics, And his network, which includes a number of people from here, have worked together and have successfully gotten about 4 grants so far, including the one I just mentioned that we heard about being funded yesterday. So that's exactly what we've done. And then the other half is leveraging and brokering access to public hospitals' information systems in order to take the ideas that these people generate and then test them in practice. So I think we've devoted, actually, when I worked it out, something like about 20% or 25% of the budget of the previous 5 years, and it's not that much different this time, is exactly about that. So for a single translational research centre, that's a really big whack of our budget. So we feel that's really important, core business. I want to say, though, that there are researchers within the informatics segments of each public hospital that are equally busy trying to understand that in their spare time. And so it's about linking with them. And we're having a meeting which I expect all of you to attend or try and get in, it's going to be limited, in July, exactly about that, about clinical informatics and the way forward for New South Wales.
Thank you. We might, with time pressing, we might draw this to a close. Let me finish with a couple of observations. David, I know you personally because we work together on the TCRN, and you are a renaissance man. You've got cancer research sorted out in your mind, you've got researchers at your disposal, you've got a TCRN that you head up, but you don't have a gift from the Australian Institute of Health Innovation. So I'd like to thank you very much.
Thank you.